Hello,
Hello.
hello, and welcome. My name is
Thank you.
Philip Lewing. On behalf of the Canadian Institute of Food Science and Technology, welcome to the 2025 Knowledge Bites Fall webinar series. Um, so I am the science director for the CIFST Food Cluster, and thank you all for joining us today. First, I'd like to thank the generous support of our corporate sponsors. And we have basically our exclusive platinum sponsor, which is Lactalis Canada, as well as we have our bronze sponsor, which is BSA Weiberg. Now, before we jump into the webinar, I just want to remind everyone 2025 is coming to an end soon, and of course, we are planning for what's happening in 2026. And of course, CIFST is planning this wonderful Canadian Institute Canadian Food Summit, which will take place on June the 10th to the 12th. And this will occur at the Hilton Mississauga in Meadowvale. So please put that in your calendar, start planning for it now, and we'd love to see all you folks and share this information with your colleagues. Before we jump into now, turning to today's session, our topic is unlocking umami, fermentation-driven enhancement of taste in shiitake mushrooms byproduct. And we're very, very happy today to have our speaker here, which is who have done most of the work in this area. It's Zeynab Razavi, who is a PhD candidate at the Food Science Department. She's a food science researcher at the University of Guelph. To start things off, let us know a little bit about our speaker. Zeynab, can you please tell us a little bit about yourself?
Sure. Hi. Thank you very much for your kind introduction, Dr. Philip. As you said, I'm Zainab. I'm currently a PhD candidate in Food Science Department at the University of Guelph, but I'm also working as a research affiliate at Agriculture and Agri-Food Canada. I'm currently in my second year of my study, and my research looks at how we can convert mushroom byproducts, materials that are usually discarded, into high valuable ingredients with fermentation. I'm mostly focused on the structural changes of beta-glucan polysaccharides because of their known properties as anti-tumor compounds, but I'm also looking at umami enhancement and flavor development. Thank you very much for having me. I'm happy to be here and excited to share my work today.
Fantastic. Now, if you all have any questions for Zainab during the presentation, we encourage you to actively engage by posting your questions in the Q&A tab. And the Q&A tab is found at the bottom of the presentation there, at the bottom of the screen. Now, you don't want to hear from me. Over to Zainab, and please share with your us with your presentation. Thank you.
Sure. Let's see. You can see my presentation, right?
Absolutely.
Awesome. Let me just put this right over here. Okay. So, today I want to talk about how fermentation driven processing can enhance the taste in shiitake mushroom byproducts. Um, Before I begin, I want to have a brief outline of today's presentation. I'll start by acknowledging my collaborators, followed by a short introduction of mushrooms and their relevance in food system. I'll talk about why shiitake mushrooms are specifically a valuable target in this research. Then I'll define umami and compounds that are contributing to this flavor. And I want to then discuss the two major challenges in the mushroom industry, which is their very short shelf life and also the large amount of waste that they're produced during production. And then after that, I want to introduce fermentation as a preservation and value adding method. And I'll walk you through the overall process flowchart of this project and the key results and finding. and then I'll conclude with the planned future work. So to begin with, I'd like to acknowledge our collaborators who make this project possible. Sample preparation and fermentation are carried out at the Food Development Group by Dr. Raj Verma and the support of his co-op student Megan. Here at AFC Guelph, we focus on developing the analytical methods and quantifying the umami compounds and also the beta-glucan from these mushrooms. We source fresh mushrooms from local farms, and once our analyses are complete, our partners at AFC Quebec and Hong Xing Restaurant will continue with scale-up and production development. So each collaborator kind of contributes at a different stage of this value chain, and I'm very grateful to be part of this team. Edible fungi include more than 2,000 species consumed worldwide, and they're widely appreciated for their natural value. Carbohydrates make up the largest portion of their dry weight, typically around 50 to 65%. And mushrooms are also a good source of high quality protein with a complete essential amino acid profile and a good digestibility. In addition, they kind of have more important bioactive compounds, for example, phenolics, antioxidants, and beta-glucans, which I'll talk about them later. And they're also very low in unsaturated fat. So together, these characteristics make edible fungi a nutrient-dense and health-promoting food group. Mushroom production is often considered one of the most sustainable areas of agriculture. Growers are sometimes calling it the ultimate recycler because mushrooms can be cultivated on byproducts and waste streams of other sectors, turning low-value materials into a productive growth medium. Mushroom farms also have a very small environmental footprint. They require low energy input and water use is extremely efficient. For example, for producing 1 pound of button mushrooms, only less than 2 gallons of water is used compared to about 50 gallons for many other fresh produced items. And also because mushrooms grow year-round, they need very little land or sunlight, and they offer a highly, resource-efficient model of food production. Shiitake mushrooms are an ideal focus of this study for several reasons. First, they are rich and known for their bioactive compounds. They contain high level of beta-glucans with health-promoting properties, and they are also naturally accumulate, in umami-related compounds. They have strong culinary and sensory value. They distinct, savory and smoky profile is widely appreciated in both Asian cuisine and global food applications. They're used fresh, dried, and in powdered forms, and they're common ingredients in soups, broths, and plant-based meats alternatives. But also, shiitake has a major commercial importance. It's the second most cultivated mushrooms worldwide after white button mushrooms, which means there's a large and stable supply chain. They also result in significant processing byproducts, making shiitake a relevant and accessible material for both research and industrial applications. So shiitake mushrooms are particularly known for their beta-glucans, which are the dominant bioactive polysaccharide in these species. These beta-glucans have 1 to 3 linked beta backbone with 1 to 6 linked branches, a structure that it's strongly associated with immunomodulatory and therapeutic effects. This beta-glucan has reported to have to support immune function, act as antioxidants, help regulate cholesterol, but most importantly, it's known for its anti-tumor properties by both improving the immune system of our body, but also having direct cytotoxic activity through cancer cells by apoptosis induction. So it's already used in several countries in injectable or supplemental medical products, particularly in Asia, which highlights the clinical relevance and commercial value of shiitake beta-glucans. Now that I've highlighted the key bioactive component of shiitake, I'd like to move to another major strength of this mushroom, which is its natural umami profile. Umami is one of the most defining sensory features of mushrooms, and shiitake in particular. It's recognized as the fifth basic taste alongside salty, sweet, sour, and bitter, and is which is primarily attributed to high concentration of glutamates, which are also naturally present in mushrooms. In Eastern cuisine, this taste has been familiar for centuries because ingredients like seaweed or mushrooms are naturally rich in glutamates, but it's also why umami often described as savory and meat-like because meat also contains high levels of glutamic acid. Later research expanded our understanding of umami by showing that certain nucleotides, for example, guanosine 5-monophosphates or inosine monophosphates for GMP or IMP, also contribute to the umami taste. Now that I've defined umami, I like to look more closely at the specific compounds in mushrooms, particularly shiitake, that contribute to the test. Mushrooms contain several classes of compounds that contribute to umami. The primary contributors are amino acids, specifically glutamic acid and aspartic acid, which provide the fundamental savory taste. Alongside these, we have the nucleotides, as I mentioned, IMP and GMP, which both significantly enhance the umami intensity. But in addition, we have other flavor-active compounds, for example, glycine, alanine, small peptides, and Maillard reaction products. In shiitake specifically, several umami peptides have been identified, which include tripeptides or dipeptides. But these peptides also contribute to the savory depth, but their umami intensity is generally weaker than the free glutamate. But the most significant impact occurs when there's a synergistic effect between amino acids and nucleotides. Research has shown that when glutamate interacts with the nucleotides, for example, IMP and GMP, the umami intensity can actually increase up to 8 times. And this happens because both glutamate and nucleotides bind to different but kind of cooperative sites in the umami receptors in our tongue, and they result in a much stronger signal. So altogether, combination of amino acids, nucleotides, and peptides give mushrooms, and specifically shiitake, their characteristics rich umami. Umami is particularly valuable in food formulation because it modifies the taste perception in ways that improve the overall flavor balance. First, umami can suppress bitterness, which happens with some cross-modal interaction at the taste receptor, but ultimately, basically, umami signals kind of reduce the brain perception of bitter compounds. It also enhances saltiness, which allows us to achieve the same salty, impression with less added sodium, which is extremely useful for designing healthier products. But also umami compounds stimulate, digestive responses. They can support appetite regulation and satiety, which is increasingly relevant in health-oriented foods. From a food product development standpoint, I think umami is especially helpful in application where, building a deep, slow-cooked, meaty flavor is not feasible. For example, ready-eat meals or plant-based foods or low-sodium products. This can be very helpful. So understanding these mechanisms will help developers design cleaner labels, reduce sodium, and kind of create a richer flavor profiles without relying on artificial enhancers. And if anyone is interested in exploring these concepts in more depth, I highly recommend the book here that I put the picture, Umami Taste for Health, because it provides kind of an excellent scientific and parallel overview of umami chemistry. Now that we've looked at why understanding umami is valuable in product development, I think it's also important to recognize one of the biggest limitation of working with mushrooms as a raw material. Mushrooms, including shiitake, have a naturally short shelf life because they have very high water content, typically around 85 to 90%. And once they're harvested, these cell structures become damaged. The internal water quickly migrates to the surface, and this leads to noticeable changes in texture and firmness. So unlike fruits and vegetables that have a protective cuticle, mushrooms lack a surface barrier, and this makes them extremely vulnerable in physical damage, microbial growth, and moisture loss. So as a result, spoilage occurs very quickly, often within 1 to 3 days in ambient temperature, or only 3 to 5 days under refrigeration. Building on the issue of short shelf life, mushroom also undergo a series of rapid biochemical and physical changes and spoilage mechanisms after harvest, which further limits their stability. Their high water content promotes microbial growth, accelerates enzymatic and non-enzymatic browning reactions, and leads to discoloration, oxidation of carbohydrates, loss of texture and firmness, change of flavor, sometimes off-flavor production. So this short shelf life is a major challenge for both producers and also processors, but it's a key motivation for developing preservation and value-adding strategies such as fermentation. But beyond their short shelf life, another major challenge in this sector is the scale of global production and the amount of waste that it generates. Mushroom production continues to grow rapidly worldwide, estimating around 45 to 50 million tons per year. And the majority of these outputs come from Asia, Pacific regions. China is the leading producer, accounting for about 75 to 90% of the global volume. But with this level of production, the industry also generates a significant amount of waste, and roughly around 5 to 20% of the total production is waste. Several types of byproducts are generated, and these include the mushroom stem waste, spent mushroom substrate, and misshapen mushrooms. And for the purpose of this project, the most relevant category is the misshapen mushrooms because they kind of contain essentially the same chemical compositions as the normal mushrooms, the same umami, the same beta-glucans, and they only differ in appearance and consumer acceptance. Misshapen mushrooms are typically produced when water content, air circulation, or nutrient availability during cultivation is not optimal. And although they kind of reduced overall commercial yield, they remain fully edible and can be utilized once they're sliced or crushed. Spent mushroom substrates is also produced in large quantities, roughly 5 kilograms of substrate for every 1 kilogram of fresh mushrooms. But for the study, the misshapen and irregular mushroom represents the most valuable byproduct stream due to their similar biochemical profile. Given the amount of waste that's generated in the mushroom industry, there is a clear opportunity to preserve and recover value from these byproducts. One promising direction is the development of natural umami enhancer ingredients. There's growing interest in clean label flavor solutions, and consumers are showing increased resistance to synthetic additives. Natural flavor components from mushrooms can provide a feasible alternative. For example, research has shown that adding 2% oyster mushroom powder can significantly improve the quality and consumer acceptance of vegetable soups. With this opportunity, one of the most effective strategies for preserving mushrooms and enhancing their flavor is fermentation. Fermentation is considered a mild, clean label technique, making it highly attractive for both consumers and also product development perspective. It helps extend shelf life while allowing microorganisms to metabolize native substrates and transform these into organic acids, and enzymes or flavor-active compounds. These microbial conversions can improve sensory attributes, particularly flavor and texture, and in some cases, they even contribute additional health-supporting effects. But selecting the right microorganisms is essential for both preservation and flavor development. In fermentation, a wide range of microorganisms can be used, for example, bacteria, particularly lactic acid and acetic acid bacteria, and various fungi, including yeast and mold, such as mucor or aspergillus species. They all play distinct roles in transforming mushroom components. In a preliminary screening by another research group, they evaluated 4 different microorganisms. And from these 4, they kind of show that Lactobacillus plantarum has the best performance in umami increase in shiitake mushrooms. So that was kind of a starting point for our project to choose Lactobacillus plantarum. In this slide, I want to summarize how bacterial fermentation enhances the key umami compounds in mushroom. Specifically, as we talk, glutamic acid, aspartic acid, IMP, and GMP. So during fermentation, Lactobacillus plantarum produces proteases and glutaminases, which break down the proteins and they can release the free glutamate, aspartate, or small peptides. And these reactions directly increase the amino acid based umami fraction. But at the same time, we have phosphatase activity, which contribute to the rise of nucleotides, for example, IMP and GMP, which are crucial for the synergistic umami enhancement. There's also the natural acidification that occurs during bacterial fermentation, which helps stabilizes flavor-active compounds. So overall, bacterial fermentation improves digestibility by breaking down these complex macromolecules, but it also introduces organic acid or flavor compounds that add the desirable complexity and richness that we're looking for. So in this project, we are aiming to optimize the fermentation condition and maximize the formation of these key umami compounds by tuning the Lactobacillus plantarum fermentation with parameters such as mushroom percentage, bacteria content, temperature, or even pH to enhance these compounds by promoting metabolic pathways that we talked about. Our process has 3 steps. We start with collecting the shiitake mushroom waste from local farms. They're then dried and milled into a fine powder to ensure consistency. The powders are rehydrated, which would become the substrate for fermentation. For the fermentation step, the solution substrate will be transferred into the fermentation vessel. Lactobacillus plantarum is introduced, and then we have the fermentation conditions. After fermentation, the samples are processed, umamis are extracted, and we will analyze these compounds with liquid chromatography or LCMS. But in our step, we have a drying step, and I think it's very important to talk a little bit about it because it's an essential step during this process. We previously discussed that shiitake mushrooms naturally contain about 85 to 90% moisture, which makes them extremely perishable. And by removing water with a heat-based, drying step, we slow down or even inhibit several degradation pathways, including the microbial growth, the texture collapse, or the ripening, and even the lipid oxidation. So we're extending the shelf life from just a few days to several months. We're reducing the weight and volume for easier storage and transportation. And we're also concentrating the characteristic of this shiitake flavors, specifically, umami and aroma. But also, drying gives us consistent, uniform starting material for fermentation experiments and for accurate analytical comparisons. Beyond preservation, the drying process also has important effects on the flavor chemistry of shiitake mushrooms. When shiitake mushrooms are exposed to heat during drying, a number of chemical transformations occur. The major one is the Maillard reaction, which is a series of reactions between amino acids and reducing sugar, and it will generate a diverse set of volatile compounds that gives umami dried powder their distinct and intensified aroma. But at the same time, heat promotes protein degradation. It will help release additional amino acids and small peptides, and these compounds will further enhance the savory and umami characteristics. To evaluate the impact of fermentation on umami, we first established a preliminary fermentation conditions. So on the results that I want to talk about, we used 5% shiitake substrate and we added 0.25% Lactobacillus plantarum. We fermented the solution for 24 hours at 40 degrees Celsius, and our sample had a final pH of 3.22. After fermentation, we centrifuged and separated the supernatant, and we extracted the umami compounds from the supernatant and analyzed them with LCMS. Before looking at the results, I'd like to briefly introduce the metric that we use to quantify umami intensity, which is called the equivalent umami concentration, or EUC. So EUC expresses umami taste intensity in terms of how much monosodium glutamate, or MSG, would produce the same perceived umami in 100 grams of the sample. It's calculated from the concentration of the key umami amino acids: glutamic acid, aspartic acid, IMP, and GMP. And the benefit of this formula is that it combines not only the individual contribution, but also the synergistic effect between glutamate and nucleotides. And overall, the higher the EUC value would be the stronger umami intensity. And this is the measure that can be used to evaluate the impact of fermentation on flavor development. Here I'm showing the effect of fermentation on the free amino acid profile of shiitake mushrooms. In this graph, we see that several key amino acids increased noticeably after fermentation. Specifically, those are linked to flavor development. Alanine, glycine, serine, they all rose significantly, and most importantly, glutamic acid and aspartic acid, which are the primary umami amino acids. These changes reflect the proteolysis activity of enzymes during fermentation, where these enzymes release free amino acids from protein-rich cell structure. But to better visualize the impact on sensory qualities, in this graph we have grouped amino acids according to their taste attributes. And here we can clearly see the shift that umami-related amino acids increased, consistent with the specific rise in glutamate and aspartate. Sweet-tasting amino acids increased, which is expected because we saw like alanine and serine increase sharply. Interestingly, bitter amino acid decreased, with aligns with the reduction, in histidine and arginine, and also tasteless amino acid decreased. So here we have, a larger proportion of amino acid pool shifted toward flavor-active molecules, and overall fermentation not only increased the total amino acids, but also reshaped their distribution towards, more desirable taste contributes, which are umami and sweetness, while suppressing bitterness. Next, we examined how fermentation affected 5 nucleotides that also contribute to umami intensity. So here we have the concentration of 5 main nucleotides in shiitake mushrooms before and after fermentation, but GMP and IMP being the main umami contributors. So GMP's level decreased, maybe because these nucleotides are quickly metabolized by Lactibacillus plantarum as energy maybe sources, but we see a significant increase in IMP, which is particularly notable because IMP is a strong umami enhancer. Here I'm showing the calculated EUC values for the control and the fermented shiitake samples. And as we mentioned, EUC could kind of give us a combined umami impact of amino acids and nucleotides in a single value. So in the control sample, the EUC was approximately 768 milligram MSG as 100 grams of dried sample, while the fermented samples showed a slightly lower EUC, about 734. So although the fermentation kind of increased the free glutamates substantially, maybe the reduction of certain nucleotides, for example, GMP, had brought the overall calculated EUC slightly lower. But it's also important to account that this formula does not take into account all the umami compounds. So at this stage, this result kind of tell us that fermentation is clearly modifying the umami profile, but not yet maybe in a way to maximize the calculated umami intensity value. And further analysis is needed. We are currently working on optimization. We are testing different parameters, including mushroom to bacteria ratio, temperature, or even pH, to kind of better understand their contributors to their contribution to umami development and ultimately improvement in the EUC value. Now that I've shown our current results and optimization work, I'd like to walk you through what is the next step of this project and how it would look like. So this project is a multi-year, integrated development pathway that involves several collaborating groups. Once we have optimized the ingredients, the food development group will explore their application in food, maybe plant-based meat analogs or maybe Asian sauces. And then the project will advance towards pilot scale production or commercial scale production, and it will allow us to test the feasibility of producing these umami enhanced ingredients at a larger volume. And then finally, there would be consumer and restaurant evaluations to assess the flavor performance in real culinary settings. I also wanted to take this opportunity and briefly highlight the other side of this project, which is the structural analysis of mushroom beta-glucans, which is a major strength of our lab in AFC. So in this part, we are examining how fermentation is affecting the structural integrity of Shiitake beta-glucans. A little bit of background. Shiitake beta-glucans are primarily beta 1 to 3 backbones with 1 to 6 branches, forming a complex network with the chitin and proteins in the fungal cell wall. The bioactivity of mushroom beta-glucans, including its antitumor or immunomodulatory effects, is highly dependent on its native structure, its molecular weight, branching pattern, its triple helical structure conformation. And one of the challenges in polysaccharide research is balancing yield improvement and structural preservation in extraction while extraction and purification, because many of these extraction techniques kind of damage or partially degrade these glucan and chitin protein matrices. But interestingly, we saw that during fermentation, and Lactibacillus plantarum can play a very interesting role here. We saw that during fermentation, because of the production of organic acids and cell wall modifying enzyme, and because the pH is lowered, it will result in loosening the cell wall network, and this will increase the beta glucan extractability without even relying on harsh chemicals or high temperatures. To understand these structural changes in depth here in our lab, we use a set of polysaccharide characterization tools in our lab. We start with looking at the monosaccharide composition with an ion exchange chromatography. We then look at the linkage pattern with methylation and GCMS analysis. And we also look at the detailed structure, the conformation, and everything with one-dimensional and two-dimensional NMR. And finally, we analyze the molecular weight with size exclusion chromatography. So these techniques have helped us to get a full structural profile of the mushroom beta-glucans extracted from fermented and non-fermented samples. And our ultimate goal is to kind of connect these structural changes to functional properties, for example, and also their bioactivity, to kind of better understand how fermentation can be used to not only enhance flavor, as we talked about, but also to modulate the functionality of mushroom polysaccharides. As a final conclusion and a take-home message, so shiitake byproducts are underused raw material with strong nutritional and sensory potential, and fermentation can transform these low-value waste into flavor-rich, functional ingredients aligned with sustainable priorities by increasing amino acids, nucleotides, and also generating complex peptides, but also improve the extractability of mushroom beta-glucans. This work supports industry-relevant innovation by creating a dual-value ingredient, aligning with clean label and sustainability needs in mushroom supply chain. And I would also like to end by acknowledging the incredible support from our collaborators. A special thanks to Food Development Group, Dr. Steve Kui and Dr. Shi Wang at AFC, Hongxing, and all the mushroom farmers who made this work possible. Thank you very much for your attention. I hope you enjoyed this presentation, and I'm happy to take any questions.
Thank you, Zeynab. That was very, very informative, a very detailed presentation you did in terms of taking us through in terms of some of the valuable things that we can get from mushrooms. I'll just jump into the Q&A. There's one question here, and the question is, based on your analysis, how high is the protein content on a fresh weight or dry weight basis? What is the protein content in mushrooms? Do you recall?
Yes, so in the dried mushroom powders, the before fermentation, the protein content was around 26%, but after 24 hours of fermentation, the protein decreased to around 6%.
About 6%.
Yeah.
Okay. So on a dry basis, the moisture content is about 5% or so.
I
The
think it's even
residual
less than that. I think after drying, we measured the moisture and it was around 2%.
2%.
Yeah.
Okay, 2%. Yeah. This is very interesting work that you're doing here because what you've done, you've combined not only the flavor changes here, but also the health implications with respect to the beta glucans. And there's another question here. How did you control the growth of other microbes during your fermentation step?
Before adding the Lactobacillus plantarum, we have a sterilization step. We use autoclavation, and then we add the bacteria.
Yeah. Okay. There was another question here. Do you know how growing conditions of the mushrooms can affect control umami of fresh or fermented product? I'll just repeat that. How the growing conditions of the mushroom could affect um, the umami of fresh or fermented product.
Um, I actually, um, because there are many factors that could affect, but I don't currently have like a good specific answer to give, like how exactly what condition would result in what. So, but I would kind of look through it, and if possible, I could, I don't know, connect to this person and respond to them because it's actually a very good point.
Okay. From the data that you presented, there was an increase in terms of the IMP, and but the GMP didn't. Can you kind of speculate in terms of? I know I'm putting you on the edge here. Can you speculate in terms of why IMP increased and GMP decreased?
I'm actually looking into that because I'm very interested to see how different conditions would result in like the change of IMP and GMP because actually I was not expecting to see like a controversial effect on that
Right.
because I personally thought if, for example, even if nucleotides would be used as, a nitrogen source, maybe we would see a similar increase in all of them, not different. But maybe, I don't know, the enzymes are more they have more affinity towards one of the amino nucleotides and less towards another.
Okay. Are there any other questions from the audience out there? Oh, there's another question. Um, uh, this is from Yvonne Yon, Dr. Yon. Um, Really interesting presentation. How will the cost of this type of product compare to hydrolysates or protein hydrolysates? Uh,
That is a very, very good question that I also, again, don't have a specific answer to that, but very good question. But I think, like in general, because, for example, there is different fermentation types to apply, and I know we could have also used solid state fermentation, but I know the controlling on solid state fermentation is a lot more difficult than solution state fermentation. So we're, I think, if we get great results with solution state, we already are using a process which is not that expensive. It doesn't need that many control conditions. But in terms of like final cost compared to others, I would need to look into that.
okay, this might be another project for you for the future in terms of doing some comparative evaluation of yeast hydrolysate, other protein hydrolysates, and shiitake mushrooms, fermented products. Yeah, there
Yeah.
you go. So that for those who are listening out there, we the CIFST cluster um, in a couple of years, we'll be sending out requests for proposals. So these are some of the things that you can be thinking of for the future in terms of how can you add on to some of the work that you're currently doing here. Anyway, I'm doing all the talking here. Any more questions from the audience? Okay, if not, Zeynab, on behalf of CIFST, thank you very, very much for the informative and well presented presentation. For the audience, this is the final presentation of our 2025 Knowledge Bite Fall webinar series. We appreciate everyone's active participation today and look forward to having you again in the new year. Uh, Please keep an eye on the CIFST website along with a newsletter for upcoming 2026 webinars. So on behalf of CIFST, CIFST Food Cluster, Zeynab, and myself, have a wonderful holiday, well rested, and thank you again and take care. Bye bye for
Thanks.
now.
Bye.